Now we get to a really important lesson about how to predict the trait for an offspring. This square right here is called the Punnett Square. You've probably seen it in 7th grade. And it is for one trait crossing with the same type of trait. Therefore, it's called a monohybrid cross. Mono means one, so we're only dealing with one trait. Here we're going to use the trait of what is called regular thumbs and hitchhiker's thumbs. I personally have a hitchhiker's thumb. I think it's pretty cool. I'll pause while you determine what type of thumb you have. Alright, so imagine that you have a mom who is heterozygous. So here, this is the symbol for a woman, so I'm going to put T and T. So these are her two alleles. That's heterozygous. We know that the big T is for the regular thumb, and so the big T is going to be dominant. Alright, so the mom is going to have a heterozygous regular thumb. We're going to say that the father, this is the symbol for male, has the same type of thing. He's heterozygous, he has a regular thumb. You can use this Punnett Square to predict the probability that their child will have a regular or a hitchhiker's thumb. So you put the letters in for the mom, the letters in for the dad, and then you're actually just going to cross them together. So, for example, this box will have a homozygous dominant, so big T, big T. This one will be heterozygous, big T, little t. This one will be heterozygous, big T, little t. And my last box is going to be homozygous recessive. So it'll have two little t's. Alright, so when I look at proportions or probability, I'm going to look for my big T, big T. So my homozygous dominant the probability ends up being 1 out of 4. Because I can see that I have 1, 2, 3, 4 different possibilities, and this is just one of them. So I'm going to put a 25% here, because that's 1 out of 4. For my heterozygous one, I can see that there are actually 2 of them that are heterozygous, which means I'm going to end up with 50% probability, because this is 2 out of 4, 1/2, 50%. And then I'm going to do the same thing with my homozygous recessive. I can see that my homozygous recessive, the probability is 1 out of the 4, which is going to give me a 25% probability. However, this is really important for you to understand that the genotypes, the percentages for the genotypes, don't necessarily match the percentages for the phenotype. As you can see here, I only have 2 different types of phenotypes, whereas I had 3 different genotypes. So I need to think about which ones will have a regular thumb. The homozygous dominant will have a regular thumb, and the heterozygous will have a regular thumb, because big T beats little t, regular thumb beats hitchhiker's thumb. I'm going to add these together, so 25% plus 50%, and I'm going to end up getting 75%. That leaves me left over with the percentage for who we actually see that has a hitchhiker's thumb, and that's going to be the people that are homozygous recessive, so little t, and I don't have to add anything to that, so that's just going to end up being 25%. So my final genotype proportions is going to be 25 to 50% to 25%. For my phenotype, it's going to be 75% to 25%, or 3, 2, 1. So now we can look at Mendels Peas and understand why the different generations turned out the way that they did. So this yellow pea in the pea generation and this green pea in the pea generation, I'm going to write in what they were, what their alleles are. So the yellow pea is going to be big Y because it's homozygous dominant. The green pea is going to be little y because it's homozygous recessive. When I cross these two together, I'm going to end up with actually all of these being heterozygous. So when I look at my genotype, I'm going to have 0% homozygous dominant, I'm going to have 100% heterozygous, and 0% homozygous recessive. When I think about the phenotype, I'm going to see that 100% of these end up being yellow, and 0% end up being green. So it's 100 to 0. Now when I look at my F2 generation, I'm going to cross 2 of these guys together. So I'm going to look at big Y, little y, big Y, little y. I'm going to cross these, and it's going to look like this. Oops, this is actually a big Y. Big Y, little y, and little y, little y. So my genotype actually ends up being 25% homozygous dominant, 50% heterozygous, 25% homozygous recessive, and my phenotype ends up being 75% yellow to 25% green, which is exactly what Mendel observed. So now we want to think about what happens if we look at two or more traits. For example, what if we want to look at both the color and whether or not the pea is wrinkled? Here we have two traits. The yellow allele is dominant and is still a big Y, and the green allele is still recessive and is still a small y. We're going to add the idea of shape to the pea. The round allele is dominant, so this is dominant, while the wrinkled allele is recessive and is shown by little r. This brings us to Mendel's second law, which is called the law of independent assortment. This states that a random distribution of alleles occurs during gamete formation. Genes on separate chromosomes will sort independently during meiosis. In other words, you're going to randomly get gametes that have either allele for each trait. We'll do the same thing as before, in which the parental generation, or P, is a P that is homozygous dominant and homozygous recessive. So we've got homozygous dominant and homozygous recessive. These cross, they make an F1 generation, and when they make the F1 generation, you're going to get one of each type of allele. So I'm going to get big Y, big R, little r. Now, when this guy goes to the next generation, so it's going to make its gametes, it's going to make 4 different gametes, each of which have either of the alleles. So for here, you can see this is big Y, big Y, small r, little y, big R, and little y, little r. All of these gametes, as you can see, then go on to be placed into my dihybrid cross square in order to determine what the F2 generation, the generation after this one, would look like. So let's try and actually do this cross. This is a dihybrid cross because it's for 2 types of traits instead of for 1 type. So if I look at this part right here, big Y, big R, cross big Y, big R, you're going to end up with a homozygous dominant for both traits, which is going to be yellow and it's going to be round. This next one is going to be big Y, big R, little r, so it's homozygous dominant for the color, but heterozygous for the shape. So this still ends up being yellow and round. Now we're just going to do that for the rest of these. So this one that I just moved, you'll see that it has heterozygous for the color, so it's still going to be yellow, but now we actually have homozygous recessive for the shape, so it's actually going to be wrinkled. I'm going to continue placing these. Alright, so now we're seeing that this is recessive for the color, so it's actually going to be green, but it's now dominant, homozygous dominant for the shape, so it's going to still be round. This next one, same kind of deal, except for the shape is heterozygous, but it still is seen as round. I'm going to place this one. I don't have that one. This one right here is going to end up being big Y, big R, little r, and that'll end up being the same as this one right here. Then I'm going to have big Y, and small r, so that ends up being wrinkled and yellow. This next one is little y, big R, that is green and round. And then this last one is homozygous recessive for both the color and the shape, so it's going to end up being green and wrinkled. For this next example, I'm going to show you another dihybrid cross. In this case, a pea is heterozygous yellow, which means it's big Y, little y, and it is wrinkled. I know wrinkled is the recessive trait, so this has to be homozygous recessive, so it's going to be little r, and it's going to be crossed with a pea that is green. I know that green is the recessive trait, so in order to see a green pea, this must be homozygous recessive. So little y, and it is homozygous round. So it tells us it's homozygous and it's the dominant gene, so it must be big R, big R. Now remember, I'm going to take one of each of these traits and put them into our gametes. Okay, so it's going to be Y, little r, big Y, little r, small y, small r, small y, small r. I'm going to do the same thing for this one. So small y, big R, small y, big R, small y, big R, small y, big R. So actually, because these are all the same, I can actually just ignore these. I'm going to ignore these, I'm going to ignore this row. And then the same here, because two of these columns are exactly the same, I'm going to cross out one of them. I'm going to ignore that one. And for this one, I'm going to ignore this one because it's the same as the other column. So now I'm actually only looking at two boxes, and I know that big Y crossed with little y, and then big R crosses with little r. That's heterozygous for both traits. That means it must be yellow and round. So let's see if I can grab one of these. All right, well, it's going to be yellow and it is going to be round. And then I know for my other trait, it's going to be little y with a big R, little r. And that is going to be homozygous for the color, so it's going to be green. But it's still heterozygous for the shape, so it's still going to be round, just like that. All right. Because I can ignore the other ones since they're just going to be repeated, my percentage actually ends up being, I end up with 50% yellow and 50% green, and they are both round. Alright, that's it for today. Make sure you go back to my digital portfolio and you answer my practice problems. If you want any more practice, you can go ahead and go to this website. Good luck, and I will see you in class tomorrow.